Hello students, now I will start with new topic that is about RNA. RNA is considered as first genetic material. As RNA is one of the nucleic acid, it is also considered as polymer of nucleotides. What are nucleotides in RNA composed of? Nucleotides in RNA are composed of pentose sugar, that is ribose, and nitrogen bases, adenine, guanine, cytosine, and uracil. And the third one is phosphate molecule. Phosphate molecule is having this double bond with oxygen and 3 hydroxyl groups. RNA, it exists as a genetic material in some types of organisms, and also it functions as a catalyst. RNA functions as a genetic material and also as catalyst. As genetic material, it is present in retroviruses, which does the reverse transcription in the host. As catalyst means in the form of enzymes. What are the forms of enzymatic type of RNA? One is ribozyme and also RNases. RNA is synthesized from DNA by the process of transcription. So, RNases are synthesized from DNA by the process of transcription. So during transcription, there is special type of enzyme called as polymerase. We also call it as RNA polymerases, which scroll over the template strand during the synthesis of mRNA, and it is majorly of 3 types, and those types are responsible for synthesis of different types of RNA. So the type of enzyme that is majorly involving in transcription is RNA polymerase. We will discuss about how this RNA polymerase is functional during the process of transcription. RNA polymerases are of 3 types. First one, RNA polymerase 1. It supports or it transcribes the genes into rRNA, or it is responsible for synthesis of rRNA. Second one, RNA polymerase 2. It is responsible for synthesis of mRNA or messenger RNA. Third one, RNA polymerase 3. It is responsible for synthesis of tRNA, and also it is considered to be responsible for synthesis of 5S type of rRNA, and also small nuclear RNA. Later we will discuss what are functions of this small nuclear RNA, RNA, etc. In prokaryotes, RNA polymerase different types are not existed. Only one type of RNA polymerase is responsible for synthesis of different types of RNA. But in case of eukaryotes, all these 3 types of RNAs are synthesized from different polymerases. In prokaryotes, only one type of RNA polymerase is present. for synthesis of different types of RNAs. But in case of eukaryotes, this is in case of eukaryotes. Next, about mRNA. The concept or the idea of synthesis of mRNA from DNA is given by Jacob and Monod in the year 1961. The concept and idea about mRNA synthesis from DNA was given by Jacob and Monod. In this chapter also, just like Principles of Inheritance chapter, scientist names and the contributed year is also important. 1961. mRNA is synthesized from DNA by the process of transcription. Transcription is also called as heterocatalysis. What is meant by heterocatalysis? Synthesis of one type of substance or nuclear material from other type of nuclear material. Means synthesis of mRNA, which is nothing but related to RNA. It is synthesized from not the previous any RNA, but it is synthesized from genetic material, that is DNA. So we are using the term heterocatalysis. Hetero means different. If DNA is synthesizing another DNA, then what is the term? The term that is used for synthesis of new DNA from the existing old DNA, the term used is autocatalysis. But synthesis of mRNA from DNA is called as heterocatalysis. mRNA constitutes of only 5% of the total RNA. During transcription, we see two different forms of mRNA. Basing on its active level, there are two types. One is heteronuclear mRNA. Another one is completely matured and active form of mRNA. So, we see hnRNA. It means heteronuclear RNA. What is this heteronuclear RNA? We said that synthesis of mRNA from DNA is called transcription. From DNA, during the synthesis, initially hnRNA is formed. This hnRNA has two different types of genes or genetic material. It has exons and introns. Exons have genetic material that can be translated into proteins. But introns does not have any genetic information for the process of translation, or the genes, which are nothing but called as introns, are not useful for the synthesis of proteins, or we can also use the term like introns are not responsible for translation. Since there are two different types of genes, we call one stage of during the synthesis of mRNA as heteronuclear RNA. Later, by the process of splicing in presence of special types of structures called as RNA protein complexes, those are nothing but spliceosomes. This hnRNA is converted into purely active form of mRNA, means messenger RNA. So messenger RNA has exons only. splicing process is to remove introns. It supports in breakage between the connections of exons and introns, is to remove introns. And we use special types of enzymes, or the body use special types of enzymes called as ligases to join all the exons and form into one complete structure called as mRNA. The genetic material is transferred from DNA to RNA or DNA to mRNA. So, we will have small diagrammatic representation regarding this. DNA, as we said, that it is an double-stranded helical form. If we unwind this, that helical or coiled form, just we will frame out two lines that represent the double-stranded DNA. One type of strand supports in giving out its own copy, means to synthesize only one type of protein structure. That is called as coding strand. mRNA means that is newly formed from the DNA. It has the same type of arrangement of nitrogen bases and also genetic information like that of only one strand of DNA. The other strand on which the new mRNA is formed is called as template strand. Means mRNA is not copying the sequence of template, but it is copying the sequence of coding strand, but mRNA is synthesized on the template strand. The newly formed, for suppose here there is sequence adenine, guanine, cytosine, thymine. Adenine, guanine, cytosine, thymine, adenine, cytosine, suppose. Now, the mRNA will be having a sequence just like the copy of one of the strand, like adenine is common, guanine, cytosine. But in case of thymine, there will be uracil. Again, adenine, cytosine, guanine, cytosine. Again, instead of thymine, uracil. Next, adenine, cytosine. So, this is how the information is passed from DNA to RNA. So, this is considered as functional or active mRNA. Now, this first part of the mRNA is called as 5' methylated cap, because the initial structure of the mRNA, it is having an methyl group, and also here there is more content of guanine also at this initial end. So we also call this part as 5' methylated guanine cap also. Now, what is this connection? The genes that are present here are not useful for coding of any type of proteins, are not involved in the process of translation. So, the genes or the codons are non-coding sequences. Now, the nitrogen bases, when they are arranged side by side, 3 in number, then we use a term called as codons. This we will discuss later. Here again, this part I am writing as, this is initiating codon. Means what type of nitrogen sequences are existed here? The nitrogen bases existed are AUG, which are responsible for synthesis of methionine. So, this type of codon is also called as initiating codon. This middle part of all gene sequences or the nitrogen bases which are termed as codons now, are called as structural genes. Next, here is located special type of gene sequences or special type of codon sequences. Here there are 3 in number. One is UAA type of codon. Next is UAG type of codon. Next is UGA type of codon. UAA codon also called as ochre. It is not ochre, it is ochre. Next, UAG is also called as amber. UGA is also called as umber. It is also called as opal. These 3 types, since here we have seen initiating codon, so these are called as terminating codons. Means they stop the synthesis of mRNA. These are also called as stop codons. These are also called as nonsense codons. Now, so by the existence of these 3 types of stop codons, mRNA structure is stopped. Means transcription is stopped by them. Again, this is also the next gene sequence. Gene or codon sequence is non-codon sequence. Gene or non-codon sequence or non-coding sequence. It is not coding for anything. So we have 2 non-coding sequences. Now, after the non-coding sequences, there is a special arrangement of continuity of adenine type of nitrogen bases which are existed more than 200 number continuously. So we call this structure as polyadenylated or polyadenine tail. Poly means many. Polyadenine tail. So this is about complete structure of mRNA. Now, just we will see if the sequence of mRNA are like this, then what is the sequence of codons on DNA? If these are the stop codons on mRNA, then what are the sequences on DNA? Instead of uracil, in case of complementary means, in case of coding strand, there is special type of codon, and in case of template strand, again the codon sequence changes. So in case of template strand, it will be uracil means adenine, thymine, thymine. Here it is uracil. In case of template strand, it will be again adenine, thymine, cytosine. In case of UGA on template strand, the sequence will be adenine, cytosine, thymine. The same in case of coding strand, how the codon sequences will be. It will be thymine, adenine, adenine. Here it will be thymine, adenine, guanine. Here it will be thymine, guanine, adenine. So these are about stop codon sequences. These are the codon sequences on the template strand, and these are the sequences, codon sequences on coding strand of the DNA. So these both are on DNA. mRNA, after transcription, it will go out of the nucleus into the cytoplasm and will does the translation process. So after translation, mRNA gets dissociated or separated from the complex where proteins are synthesized. Moves out of nucleus into cytoplasm and does translation. Translation means synthesis of proteins from RNA. During the translation process, 3 types of RNAs rRNA, tRNA, mRNA, all the three are involved in cytoplasm. And after translation or after protein synthesis, mRNA is dissociated. Means no longer the complete structure of mRNA is existed again. Again, new mRNA has to get synthesized from nucleus, means by the process of transcription, or broken down, we can tell. So mRNA is called as short-lived. mRNA is short-lived because after the process of translation, again mRNA will be cut into small nucleotide bits. Next, about tRNA. tRNA, its full form is, so we did not mention at any place mRNA full form, that is messenger RNA. It transfers information from DNA from the nucleus into the cytoplasm. So it is messenger RNA. Then about tRNA. tRNA full form is transfer RNA. tRNA is about 15% out of total RNA. Where it is synthesized? It is synthesized in the nucleosome or the chromatin material, and then it is getting transferred into cytoplasm. It is synthesized in the nucleosome or inside the nucleus or chromatin material, and is transferred into cytoplasm. Then later it supports in the process of translation. tRNA is synthesized from the type of polymerase. What type of polymerase is responsible for synthesis of tRNA is RNA polymerase 3. Next, tRNA, it is existed in different forms. Like mRNA, before complete structural formation, we used a term like hnRNA or heteronuclear RNA. In the same way, tRNA is synthesized or is existed in 3 different forms. So during synthesis of tRNA, the first one is primary structure of tRNA. Primary structure of tRNA has one continuity long linear type of chain-like structure. So what are the sequences? There are nothing foldings, means no foldings in the RNA. It will be like a long linear strand. So the sequence is 5' end, next is D arm, next is anticodon loop, next is T psi C loop, next is amino acid binding, that is 3' end. We also call it as CCA end or amino acid binding arm. 5' end. This arm is also called as acceptor arm. This complete long linear strand in the next type of tRNA, means in the next stage, it will undergo some types of foldings, and the same structure now it is converted into 2-dimensional structure that we call as clover leaf form of RNA. 2D structure of tRNA. It is also called as clover leaf model. It is also called as secondary structure of tRNA. This secondary structure of tRNA has structures like this. This is 5' end. This is D loop or D arm. We will see complete structure later after this basic terms. This is called anticodon loop. This is called T side C loop. This is called variable arm. This is 3' end where amino acid binds during the translation process. Here there are special gene sequences, that is CCA codon sequence. We have to read this sequence of the codon not as ACC. We should read as CCA, because in a genetic means any strand of either DNA or RNA, we have to read the gene sequence or codon sequence from 5' to 3' end. So we are calling it as 3' end of tRNA has CCA codon end or codon sequence. Now, third type of structural form of tRNA is 3D structure, which is also called as inverted L shape of tRNA. So here we can write zigzagly arranged structure of tRNA like this. So it looks roughly like inverted L-shaped structure. So as we have written here, like this is anticodon, this is D loop, and this is T loop, this is variable arm. This is also called as acceptor step. Now, here will be anticodon loop. Here will be D loop. Here will be location of T side C loop. So the acceptor stem which is present here will be shifted to the top position. So this is acceptor stem. And on the acceptor stem, the bottom part will be of 5' end. The upper part will be of 3' end. So this is how the 3D structure is existed, and variable arm will be somewhere here. Variable majorly supports in amino acid recognition during the translation process. Its function is amino acid recognition during translation. Now, function of anticodon is reading the codon sequences of mRNA during translation. So it reads the codon sequence that are present on mRNA. T side C loop. This one is supporting in attachment. T loop is also termed as attachment loop. It helps in recognition of the ribosomes that are present on the mRNA during the process of translation. So T side C loop. It helps in recognition of ribosome, large subunit, during translation. Now, the function of variable arm is unknown. Another one. Amino acids bind on 3' end during translation. Here, I have used the term translation many times, and I am also using the term largest subunit, etc. So, for suppose, we will see how the structural arrangement of RNAs during translation. If you think that this is mRNA, ribosomes are having two subunits. One is smaller subunit and another one is larger subunit. So, smaller subunit will be on opposite side to that of larger subunit. Now, tRNA, somewhere it is bringing amino acid from the cytoplasm and it has to come and donate the amino acid here. So, it has to recognize the larger subunit. So, tRNA, if it has brought the amino acid here, now we are seeing in the total structure as mRNA, ribosomal RNA, tRNA as a complex during the process of translation. So that I have written here larger subunit during translation. We will read about this complete translation, how the complexes are formed, how tRNA is bringing amino acids, etc. This D loop is made up of 8 to 12 base pairs, nitrogen base pairs. T size C loop, it is made up of nearly 7 base pairs. Now, we are telling anticodon. For suppose here, if we take one of the codon sequence on mRNA as AUG, we will take AUG codon sequence we will take on mRNA. Now, what are the anticodons that are present on tRNA? That will be complementary UAC. So now these UAC are considered as the anticodon sequence that are present on tRNA. These are also made up of 7 base pairs. Complete anticodon loop, it is made up of 7 base pairs. General meaning of adapter is connecting two different parts. So during the translation process, this tRNA is connecting ribosomes and also it is connecting the amino acids, ribosomes and also the mRNAs. tRNA, also called as soluble RNA, because it is soluble in 1 molar NaCl in sodium chloride. tRNA, also called as adapter. I am not using the term O, I am using the term A here. Adapter RNA, because it binds amino acids, brings amino acids and supports in the binding to ribosomes areas, because there are 3 different areas on the larger subunit of ribosomal RNA or ribosomes, that is amino acid binding initiating or locating place 1, next peptide bond forming place, again that is also located in larger subunit only. The third place is after formation of amino acid, the tRNA will be removed. For suppose, if this is the larger subunit of RNA, amino acid initially will be brought by tRNA. In the next step, already previously existed AUG, that is methionine amino acid will be there. That will form and this is shifted to the next P site. We call it P site. This is called as A site. So when this amino acid has come near to the methionine, which is existed one, so it forms a P site, supports in the forming of peptide bond. After formation of peptide bond between the second amino acid and initially existed methionine, the tRNA again will get separated from the area called as E site, that is called as exit site. rRNA. rRNA full form is ribosomal RNA. Ribosomal RNA, how much percentage or how much amount it is present out of the total types of RNAs? It constitutes about nearly 80% of total RNA. tRNA is considered as the smallest RNA, but so we'll write tRNA is small RNA among all the types of RNAs. Next, tRNA is also, I have seen in some books how many number of amino acids or nucleotides are present here. In an tRNA, one single tRNA, it is supposed to be 73 to 94 nucleotides in one single tRNA, and it is the smallest one. So rRNA is largest RNA. Largest RNA. Next, rRNA supports in protein synthesis. Next, rRNA is also considered as the most stable RNA. Most stable RNA, because we have seen mRNA after translation gets dissociated. tRNA is the smallest one. Means after its function again it is going back in an inactive form into the cytoplasm. So rRNA is the largest, and also we can use the term longest also. Longest RNA, largest RNA, and it is supporting in synthesis of proteins. And this rRNA is having the genes that are responsible for synthesis of ribosomes. So ribosomes, when they are present on the endoplasmic reticulum that are existed on the cytoplasm, they help in synthesis of proteins. This rRNA is majorly synthesized or in nucleolus of nucleus. What is this nucleolus? Nucleolus is considered to be a dense material that is present as a separate material inside the nucleosome. A dense material that supports in synthesis of rRNA